Hello everyone. Welcome to this continued series of lectures in Biotechnology Paper 2 Biochemistry. The topic of today's discussion is amino acids and uh, amino acids classification and structure. Proteins are the main agents of biological function. They have a very important role in catalysis. They work as enzymes, say for example, enolase in glycolytic pathway or DNA polymerase in replication. And transport, proteins have very important role in transport, whether it can be lactose permease or hemoglobin in the transport of oxygen in the blood. Structural elements like collagen, keratin of the nail, hair, feathers, horns, etc. And movement is only possible due to myosin and actin of the muscle fiber. So proteins are the main working forces. Uh, Protein structure and protein function has something very important to do that we will see in the forthcoming classes. Now let us move on to uh, amino acids. Amino acids are the building blocks of protein. Why I define protein is proteins are the one of the important polymer, and the building block of proteins is amino acids. Now let us move on to the amino acids. Proteins are linear heteropolymers of alpha amino acids. Amino acids have properties that are very well suited to carry out a variety of biological functions, like capacity to polymerize, useful acid-base properties, varied physical properties, and varied chemical functionality. So proteins, amino acids are very well suited to catalyze a variety of functions. Amino acids share many features differing only at the R substituent group. When we look at the general structure of an amino acid has a carboxyl terminal and amino terminal and a R group. At the center, it has a C alpha, which is uh, which is chiral, and possibly we can expect some isomers if we have chirality. So in all the amino acids, we have around 20 standard amino acids. There will be a carboxyl terminal and there will be amino terminal, and what is going to vary is only this R group is going to vary. So depending upon the varying of this R group, we have different classes of amino acids. Amino acids have three common functional groups attached to the alpha carbon atom. The alpha carbon always has four substituents and is tetrahedral, except proline except proline, all an acidic carboxyl group connected to the alpha carbon atom, a basic amino group connected to the alpha carbon atom, and an hydrogen connected to the alpha carbon. The fourth substituent R is unique in glycine, the simplest amino acid. The fourth substituent is also hydrogen in case of glycine. Hence, we can't expect isomerism in glycine. All amino acids are chiral except glycine. So that's what I was saying. It is not possible to have chirality in glycine because we have 2H on the other side. So this is the general structure of L-alanine and D-alanine, where we can expect the mirror images, L-alanine and D-alanine, right? Amino acids can be classified depending upon in five basic groups depending upon the R group. We have non-polar aliphatic amino acids, which are 7 in number, aromatic amino acids that have R group substituted by aromatic groups. They are 3 in number, polar, uncharged, positively charged, and negatively charged. So altogether we have 20 standard amino acids. So these are the structures and classification where we can just see that the skeleton is going to remain same. The C alpha, we have the carboxyl terminal, we have the amino terminal. As I said you, in glycine we have the R group is also H. So alanine, we are having CH3 as the R group, and valine, this is the R. So the entire R is being mentioned is shown in blue color, the remaining entire skeleton of the amino acid is going to remain the same. So there will be a carboxyl terminal, there will be an amino terminal, and there will be a hydrogen atom, and you have a R group, and this R group is going to vary. So in aliphatic, so all these are aliphatic groups, hence we call glycine, alanine, valine, lysine, methionine and isoleucine as non-polar aliphatic R groups. And now let us come to positively charged groups. So lysine, arginine and histidine, they have the R group is positively charged. Hence lysine, arginine and histidine are positively charged amino acids. Similarly, we have R group that are negatively charged like aspartic acid and glutamic acid, or we can also call it as aspartate and glutamate, because you look here the negative charge extra. And we also have non-polar aromatic amino acids. So when we look at the aromatic R group here, the R group is having a aromatic ring. So hence we call these amino acids tryptophan, tyrosine, they all are aromatic in nature. Similarly, polar uncharged groups. So here these amino acids are polar, but the R group is uncharged. So this blue in color, serine, threonine, cysteine. We also have uh, 1 second, I could not read this out. Cysteine. All these are polar uncharged amino acids. Yeah. So these are aliphatic groups: glycine, alanine, proline, valine, leucine, isoleucine, and methionine. And we have aromatic phenylalanine, tyrosine, and tryptophan. We also have polar uncharged serine, serine, threonine, cysteine, asparagine, and glutamine. So these are non polar uncharged R groups. And we also have positively charged R group, lysine, arginine, and histidine.